All right, well, we are live again here. This is the CNHP seminar series. Good morning, good afternoon, wherever you're attending from. I'm your host, Rob Shore. I'm a zoologist with the Colorado Natural Heritage Program. And for those of you who haven't attended in the past, let me at least briefly take you through all of the options that, or at least all the programs that the Natural Heritage Program is participating in. And is everybody able to see my screen now? No? Let's see, let's check this. Everybody seeing seeing a beautiful scenery right now? Excellent.
Yeah.
So the Colorado Natural Heritage Program, like all the other natural heritage programs in existence, started around the 1970s as a tool for the Nature Conservancy to figure out where the last places and the last great places were. And these were outreach and survey teams that would go out and try and diagnose where the best aggregations of some of the rare, unique species were in that particular region. That has evolved since the 1970s, and now we're under the parent organization NatureServe, and we're all the 50 states, all the provinces, and a host of Caribbean and South American countries. And we still function that tool in some regard, but our premise for everything that we provide has changed slightly in that we don't just focus on rare species. We help land managers that are partners identify resource needs that they might have and help them identify ways that they want to manage for those. So I'm going to stop sharing that screen and give you introduction to our speakers today, because I'm pretty excited about today's talks. Also, forgive me if you happen to hear my co-host in the background. This is my co-host Bella, the bulldog, sleeping on the couch behind me, so it's not me snoring. But I'm really excited about today's presentations because as a zoologist, I get really excited about studying rare animals, but I'm always fascinated about how botanists understand the population ecology of plants. And so we have three experts on with us today. Jill Handwerk is our lead botanist at CNHP, and she's worked extensively with threatened and endangered and candidate species in Colorado. She has a BS in horticulture from Colorado State University and a master's in agriculture from Cal Poly in San Luis Obispo. Jill has been the principal investigator on numerous botanical research projects for U.S. Fish and Wildlife Service, U.S. Forest Service, Bureau of Land Management, and Colorado Parks and Wildlife. She's the lead author on the Rare Plant Addendum to the 2015 Colorado Wildlife Action Plan and the principal editor for plants in the Bureau of Land Management's 2015 Climate Change Vulnerability Assessment. Jessica Smith is a botanist focusing on Colorado's rare plants, conducting plant population monitoring and assessments of natural areas across the state. She has 20 years of experience assessing plant communities and populations in a variety of ecosystems around the country, with 8 years specific to the Colorado flora. Jessica holds a master's degree in marine, estuarine, and environmental sciences from the University of Maryland. Neal received his BS in Ecosystem Science and Sustainability in 2018 here at CSU, and his master's in Forest Sciences in 2020 from CSU. His master's research focused on comparing LIDAR and unmanned aerial systems. And yes, he's going to explain all these acronyms to you for forest structure characterization. Neal's recent work has included using unmanned aerial systems for rare plant mapping, as well as working as a project team lead for NASA's DEVELOP, using satellite imagery to map 2020 Colorado wildfires and evaluate forest fuels treatment's effectiveness. He's passionate about remote sensing, LIDAR, and all these fun acronyms that you're going to learn about today, point cloud analysis, and natural resource monitoring. He's also exploring photogrammetry, and he's going to definitely put in a host of terms that are going to be difficult for me to pronounce, and 3D modeling in his free time, and enjoys running and biking around Fort Collins. All right, so we have three experts on here. Before I even let them go, though, I always want to remind everybody that CSU is part of a bigger system and a bigger history. And Colorado State University acknowledges with respect that the land CSU is on is the traditional and ancestral homelands of the Arapaho, Cheyenne, and Ute nations and peoples. The area of CSU was also a site of trade, gathering, and healing for numerous other Native tribes. We recognize the indigenous peoples as original stewards of this land and all the relatives within it. As these words of acknowledgement are spoken and heard, the ties nations have to their traditional homelands are renewed and reaffirmed. CSU is founded as a land grant institution, and we accept that our mission must encompass access to education and inclusion, and significantly, that our founding came at a dire cost to Native nations and peoples whose land this university was built upon. This acknowledgement is the education and inclusion we must practice in recognizing our institutional history, responsibility, and commitment. All right, with me out of the wavelengths, I'm going to turn it over to our hosts.
Thank you, Rob. I'm Jill Handwerk, and as Rob said, I am the team lead for the Botany and Vegetation Ecology team here at the Colorado Natural Heritage Program. And I'd like to start us off with just an overview of what our team does and a little bit about Colorado's flora. So, Neal. So, what does our team do? Our core functions really are our rare plant and plant community database and the surveys we do on the ground to support our land management partners. We also conduct rare plant monitoring, habitat modeling, climate change vulnerability assessments for our rare species and some of their habitats. And we occasionally do some weed research and mapping. So, Colorado's flora. There's approximately 3,000 species in Colorado's flora, and that includes some non-native species as well. But of those 3,000, 585 plant species are tracked by the Colorado Natural Heritage Program, and that means that they're considered rare or vulnerable in the state. And of that suite of plants, 117 are also vulnerable or globally imperiled. So Penstemon yampaensis here is an example of a globally imperiled plant, only known from Colorado and a little bit of Utah. Colorado also has 15 plant species that are listed under the Federal Endangered Species Act. And there's a photo here of Sclerocactus glaucus. It's globally imperiled, known only from Western Colorado, and it is listed as federally threatened. So what's in our rare plant database? Well, it has about 14,000 mapped locations of all those 585 plant species. And that may sound like a lot, but really they occupy less than 5% of the surface area in Colorado. And our database with those sites, those mapped sites, is used to inform best management practices for our land managers. We've also developed a rare plant guide, which you can see a sample of here, and that's available on our website. And our data is also used to host an annual rare plant symposium for both professional and amateur botanists who get together every fall to talk about what's the latest in rare plant conservation and research. And rare plant surveys. This is our bread and butter here, in addition to our database. But the focus here is generally on federally listed and sensitive plant species. We assess population size and condition and refine mapped locations from points that you see here to polygons. And in so doing, we can really support habitat conservation and land management decisions when our partners have precise information about where plants occur and what condition they're in. So next we're going to talk about rare plant monitoring, and I'm going to turn this over to Jessica now, and she'll dive into some details and examples of what we do to monitor rare plants once we know where they are. Jessica?
Thank you, Jill. And I apologize, I know my name says Jill Handwerk. We had a little trouble with the Zoom, so I'm on Jill's link. But I'm Jessica Smith, and I'm going to walk you through some of the things that CNHP does for rare plant monitoring. The work that we do is done to assess habitat conditions and demographic trends of imperiled species. We monitor vegetation changes over time in response to management activities, And our results guide land management decisions and help with species conservation. Next slide, Neal. So I'm going to discuss our traditional monitoring. And what I mean by that is actually having researchers on the ground, usually counting plants within plots. And 3 of the species we're going to highlight are ones that we've been working with a lot over the last several years. Ipomopsis polyantha, which is the Pagosa skyrocket. Physaria pulvinata, that's the cushion bladder pod, and that's the plant you see pictured in this slide. And then Penstemon debilis, which is parachute penstemon. Okay, Neal. So I think we'll start with Penstemon debilis, parachute penstemon. That's the picture that you see here. That's the plant. And CNHP has had a monitoring plot, which we established in 2015 and have monitored annually. And we've had a bit of a twist on traditional monitoring with this plant. As you can see, the habitat for this plant is very steep shale slopes. And we've been working to minimize surface disturbance and having researchers walk on these slopes. So we've been using both photos and ocular observations to actually monitor the plants. This is work that's been conducted on private property with funding by state and federal partnerships. And in this photo, you can see the setup that we use. We have a tripod a camera is mounted on a tripod, and we take a series of photos of the slope. And then we use them. And this is an example of what we do. We have this on the computer, and then we zoom in and we mark the plants with, you can see the plants that are marked with the purple circles, and then we actually number them, and then we follow them through time. So the second plant we'll talk about is Physaria pulvinata, the cushion bladder pod. And CNHP has had monitoring plots established since 2016, again, monitored annually. The majority of this species is found on state lands in southwestern Colorado, and we have enough plots on the landscape that we can get range-wide population trend monitoring. Yep, and that's what our monitoring plots look like. Whoops, too quick. All right, and the last plant I'll talk about is the Pagosa skyrocket, Ipomopsis polyantha. And we've had monitoring plots established for this plant since 2017 in cooperation with the state of Colorado. In 2020, we have had monitoring expanded across the species range. I added 4 monitoring plots with funding from the U.S. Fish and Wildlife. And this monitoring work has been done to aid in state land management and also support the federal recovery plan. Yeah, so in this slide, I just wanted to give you a feel for what monitoring looks like on the ground. You can see researchers and plots. We're counting plants. The flags just help us mark what plants we've already counted, because in this case, the skyrocket is very numerous. We've monitored in subsets of large plots, and then we've also used small plots around the range. Around the property. And I do a power analysis in the field to make sure that we get enough samples to meet the precision that we want. And in this last slide, I wanted to give a shout out to our honors undergraduate student, Emily Fitzjohn, who was out here a couple years ago helping us to monitor the Pagosa skyrocket. Rob is actually her advisor. She used a technique that has been used quite a bit in wildlife sampling called distance sampling, but it hasn't been tried out with plants as much. So we were testing out a novel method. And now I'm going to turn it over to Neal to talk about meeting some of the challenges with rare plant monitoring.
Awesome. Thanks, Jessica. So yeah, just to dive in here, while there are many rare plant species that live in accessible places, some don't. And Penstemon debilis is one of these species. It really likes these super steep shale cliffs, and it seems to be pretty happy there. However, monitoring and surveying for Penstemon debilis is really challenging, since the shale is really slippery and the cliffs are so steep. It makes it really hard to find new populations, since there are suitable habitat areas just out of reach, kind of like in the image in the right, unless you have like a helicopter or you really like rock climbing. And so where my experience with rare plant mapping started, summer of 2017, I was out with Jill in Montrose, Colorado, surveying a rare plant. And she had mentioned the Penstemon debilis problem in Rifle, Colorado, and I thought it was a really interesting and perplexing question. really got me thinking about how we could try and improve some of these methods. And so eventually, after lots of brainstorming sessions with Jill and others at the CNHP, we thought it would be a really cool idea to try and use drones for mapping rare plants. They can go where people can't. They're really cheap to insure and replace. So could we use them to map rare plants? So to provide a little bit of background about unmanned aerial systems, aka drones, they can fly for about 15 to 25 minutes, and they're typically flown below 400 feet above ground. They can capture really fine resolution images like sub-centimeter, and they provide photos that are geotagged, which gives them precise location data. So here's a quick timeline of the different drone-based rare plant research that I've worked on in the past few years. So in spring of 2018, we tested an idea. Summer 2019, we applied the idea, and then summer of 2020, we scaled this idea up. So spring 2018, testing the idea. During my last semester of my undergrad, I worked with Dr. Michael Falkowski and the Colorado Natural Heritage Program to investigate the feasibility of using drones for mapping a rare plant in Fort Collins, Phaceria bellii. It's endemic to Colorado, and it's listed as an imperiled species. It has bright yellow flowers, which arrive in like late April, early May, and we really thought that that distinct coloring would help us pick it apart from other plants in the landscape. And so the similar characteristics of its habitat, this shale landscape, provided a really great place for us to try and explore some of these potential mapping methods to eventually try these for mapping Penstemon mandibulis in Rifle, Colorado. And so the secret behind using drones to map rare plants is photogrammetry. And to boil it down, it's effectively using photographs to make measurements. So in my senior year of undergrad, I kind of discovered photogrammetry technology, and I worked through the steps to apply this for drone mapping. But step one is effectively to capture tons of overlapping images of the rare plant habitat. The second step is to find these matching points between images. The third step is to stitch all the images together using those matching points. And then finally, to generate 3D models and orthomosaics from these images. And so here's a quick video that I worked on for spring 2018. So here's the plant, Phaceria bellii. Here's one of the drones that I used for the study, DJI Inspire 2. And so my study area was in Wellington, Colorado. And here's a quick example of some of the habitat. I tried a couple different methods. One of them was taking these video transects where I set up a 1 meter transect and flew the drone over the transect, capturing video to look at later. The second method was just taking a ton of overlapping photos of the shale outcrops themselves. And so I flew the drone in this serpentine pattern, capturing imagery of this of the shale. And so here's an example of one of those Phaceria captured by the drone flying right above it. So once I had captured all my imagery and taken it back home, I then started processing my imagery. And so this is those steps that I talked about earlier, where we stitch all these images into a composite orthomosaic. Then from this composite orthomosaic, we also get a cool 3D model, which is fun to look at. But in the ortho, we go and we zoom around and we look for these plants and manually mark them with a point. And this method seemed to work really well for what we were looking at. And so based on these methods in 2018, we found that it worked really well for these flat shale. So we wanted to see if we could try and apply the same method for mapping Penstemon mandibulis to see if it would be feasible for larger areas and steeper slopes. So in July 2019, Jill and I traveled out to Rifle, Colorado, and we tried these same methods as in 2018, flying super close to the shale and capturing as many overlapping photos as possible. So we flew for 1 day, we had 10 flights and captured about 5,000 images. And we got bit by a lot of gnats too. Yeah, we were mapping in an area that had known Penstemon mandibulis occurrences. So this is our study area. It's from western Colorado, above the towns of Rifle and Parachute. And these were located on top of the Rowan Plateau in the Green River Shale Formation. So when I'm flying the drone, I typically fly between 4 and 8 meters from the side of the cliff face. And I get this cool live camera view from the drone so I can see what the drone is seeing while I'm flying. And circled in red here are examples of new Penstemon mandibulis occurrences. it can be really challenging to try and see these while I'm flying. So I usually take a ton more pictures than necessary. And then I look back through these images on a laptop once the drone lands. So once we capture all these imagery, we process them to make these big orthomosaics. And they're super fine resolution, like 1 millimeter to 5 millimeter per pixel. And these really big, they're around 120 to 400 gigabytes. So they're really big data sets. But this resolution really allows us to identify these flowering plants. So like in the image you can see here, and this is one of the main features that helps it stand out from other species, are the pink-white flowers. And so with the help of student interns in QGIS, we visually searched these orthomosaics using a 1 by 1 meter fishnet, checking each cell for plant occurrences. The work study students would then mark these plants with a point, and then I would go back through and QC their work to make sure that what they found was actually a penstemon. So at our first test site in 2019, we found 1,015 confirmed flowering penstemon debilis. At our second test site, we mapped out 887 flowering penstemon. And while this manual searching method did take a long time, the results were super promising. And we decided that this method worked really well for mapping areas with known plants. But the question still remained, like, what about finding new populations that no one had ever seen before? And so that's what 2020 was all about. So we were focused on scaling up this method that we tested in 2019. And we had two primary goals for our objective: to survey new populations and to monitor existing ones. For surveying new populations, our goal was to try and explore some of this inaccessible habitat, trying to keep botanists safe from cliffs. They often wander a little too close. And we wanted to try and find some new plants. For monitoring our existing populations, our goal was to try and reduce some of the shale movement, detect the spatial positions of some of these individual plants, in order to try and help track plant locations through time. So brief overview of our methods here. First, we identified our study areas. Next, we captured our images with the drones. And this consisted of about 80 flights, covering just shy of 600 acres. We also captured about 40,000 plus images, using both red, green, blue, and multispectral cameras. Next, we processed our images with Agisoft Metashape to generate 7.5 terabytes of orthomosaics and other files. Next, we manually identified the plants in the orthomosaics, just like in 2018 and 2019, and then we summarized our population extents. So first, we explored some of these drone surveying methods, so exploring for new populations. When selecting our study areas and looking for suitable habitat, we were looking for a very specific band of shale known as the Mahogany Zone of the Parachute Creek member of the Green River Shale Formation. In this band of shale, it has similar characteristics to occupied penstemon debilis habitat, and the marking in red kind of shows examples of this band of shale. We identified these study areas through GIS and field reconnaissance, but once we had identified areas that we thought were promising, we got to work. So once we found a good spot that we thought was really promising, we would get out the drone and walk to the cliff face at the top. I would then launch the drone and take hundreds of overlapping photos of the cliff face. So once we had all these photos, we take them back to the office and process them. And so once I process the imagery, it generates a 3D model and an orthomosaic from these overlapping images. And this is an example of a 3D view of this cliff face. And this was one of our new areas that we found some penstemon debilis at. And so from all of the surveying that we did, we had some quick statistics for you. We surveyed 12 new suitable habitat locations. And from that, we found 936 flowering penstemon debilis. We looked at about 12 acres of close range orthomosaics. And this took approximately 224 hours, which is quite a bit of time. Effectively, we were getting about 219 square meters of orthomosaic per hour. And overall, our results showed us that this method worked really well for exploring previously inaccessible habitat, even though it still was time consuming to capture the imagery, process it, and then have our work studies analyze the data. So next, I'm going to talk super briefly about some of our drone monitoring. So re-examining some of these places where we know penstemon debilis are, and we're just trying to keep track of the populations. So one of our main objectives for monitoring is to try and determine if we can use drones for this long-term monitoring of populations. So we decided to test the same method that we used for drone surveying, but to see if we could apply this to a known area to see how many plants we could find compared to field surveys. So early in the presentation, we briefly mentioned that there are some new methods for monitoring existing populations of Penstemon debilis. And so in the top left image, this is one of those terrestrial photos for a quadrat. And this is captured from the ground using a normal handheld camera on a tripod. And so we take multiple sequences of these images vertically of this cliff face, and then we stitch all these images together using a different program called Microsoft Image Composition Editor, or Microsoft ICE, as seen in the bottom left photo. So it's this TIFF file, it's a big orthomosaic, and we can zoom in and out and visually inspect the quadrat for Penstemon debilis occurrences. But we were really interested to see if we could use our drone to capture images from above the cliff to kind of monitor these same Penstemon debilis occurrences in a similar way. So for testing drone monitoring, we looked at a couple of things. We evaluated 2 different cameras and different flying altitudes. And our main question was, does the resolution of the image affect how we can detect plants? While flying higher covers more ground, it results in a coarser resolution of the image. Similarly, flying lower covers less ground, but results in a finer resolution. So we also tested 2 different cameras, a red, green, blue camera, just like a normal one on your smartphone, as well as a multispectral camera. And the RGB camera has a super fine resolution, so it's pretty easy to detect our plants. The multispectral has a coarser resolution, which makes it harder to detect plants, but it also has more spectral channels that we can kind of see if there's any sort of distinct characteristics of Penstemon debilis in other wavelengths of light. And so our idea here was to try and use plant locations found in the red, green, blue photos to try and train our multispectral orthomosaics for more automated spectral detection. And so the multispectral camera was a 5-band camera. So for each image capture, we captured 5 separate images, a red, green, blue, red edge, and near infrared image. On the left, you can see the example of the multispectral camera, and on the right, you can see it in action flying above our cliff. And so we stitch all the red, green, blue, and multispectral images together into these georeferenced orthomosaics. So after we stitched all these images together, we then manually lined up those terrestrial photo quadrat locations, like the ones from the images, on top of the drone orthomosaics. And you can see those quadrat locations in red here. We then manually went through and found plants within the 2 red, green, blue orthomosaics, the ones on the left, and we found plants within each of the quadrats, and they have different spatial resolutions. On the right, we have our multispectral orthomosaic overlapping with the red, green, blue orthomosaics. And so we found that flying altitude and the spatial resolution really mattered for detecting Penstemon debilis. As you can see on the image in the upper left, it's very easy to make out Penstemon in this image. And this was flown around 4 meters in altitude with the red, green, blue camera. In the picture on the right, it's much more difficult to see that this plant is a Penstemon. And this was flown at 13 meters in altitude with the same exact camera. In the bottom picture, we have the same plant from the multispectral orthomosaic, which is tons harder to identify than the red, green, blue images, just because of the resolution of the camera. And this one was flown at 8 meters in altitude. We compared our photo quadrat plant occurrence data to our 1 millimeter and 3.4 millimeter orthomosaic occurrence data using a Wilcoxon rank sign test to try and determine if these 2 samples were statistically different. So we found that the 1 millimeter and the photo quadrat data were not statistically different, but we did find that the 3.4 millimeter and the photo quadrat data were statistically different. And to try and visualize these trends, we generated linear models comparing the photo quadrat and the drone monitoring data. So the drone monitoring data is on the x-axis and the photo quadrat data is on the y-axis. And the 1 millimeter had a really strong relationship with a nice R-square of 0.92, but the 3.46, the R-square is just not quite there. Yeah, and so we just kind of effectively determined that that 1 millimeter resolution was way better for doing monitoring compared to the 3.4 millimeter. And so when we were assessing our drone monitoring methods, we realized that we had a lookalike species at one of our study areas. So in the figure on the left, we have this Astragalus lutosus with the yellow flag. And on the image on the right, we have a Penstemon debilis by the orange flag. And these look so similar when you're looking at them in the drone orthos, and it's really hard to differentiate between the two. So we used our RGB locations to sample pixels from the multispectral orthos and for both species. And so once we had enough spectral samples, we tried comparing the spectral signatures between these two plants. And so as you can see in the spectral signature graph, Penstemon debilis and Astragalus look super similar in red and blue. And this indicates that using an automated spectral identification might also pick up some Astragalus lutosus individuals. So we'll need to be really careful with this method moving forward. We're still kind of exploring some of these potential approaches for automated classification, and efforts are ongoing to try and tease these two species apart. Overall, there are a number of pros and cons for using drones for air plant monitoring. Some of these pros include that we could locate plants behind bushes and vegetation that aren't quite visible in the photo quadrats. It also takes less time to capture the images for the drone compared to our terrestrial method. And we also have these georeferenced plant locations, which really helps with tracking population trends over time. And it was really cool that the drone didn't disturb the delicate shale at all, compared to a little bit of disturbance from the photo quadrat method. Some of the cons include orthomosaic blurring. We had a couple of shadow and lighting issues from our image capture times with the drone. And the resolution of the drone imagery was coarser than the terrestrial photos just because of the nature of the camera on the drone. And the drone monitoring orthomosaic processing time took quite a bit longer than processing the terrestrial photos. So here, one of our pros for drone monitoring is that we get these georeferenced plant locations. So in the figure on the left, we have the photo quadrat with the plant location circled in red. And in the figure on the right, we have the UAS orthomosaic with those same plant locations demarcated with the red dots. And the orange dots are actually the plants that we found in the UAS orthomosaics before we compared where the true locations were. So in this case, we found all of the plants in this orthomosaic compared to the terrestrial photo quadrat method. So some of the cons, we have this crazy orthomosaic blurring, as you can see in the figure up in the upper left. It makes it really hard to see what's going on there. In the figure on the right, we have some really blurry pixels that we're just not quite sure what that is in the picture. And then in the figure in the bottom, we have some of these lighting changes where we were flying early in the morning. And as we progressed towards later in the day, we had some differences between the images and it made it kind of hard to see what was going on. So some of our upcoming work, we're continuing with this multispectral classification method, trying to further explore different ways that we can tease this data apart and really utilize all that spectral information that we have for some of these monitoring areas. We're also planning on mapping some new Penstemon debilis populations this upcoming July 2021. And we're going to try and continue to refine some of our rare plant monitoring methods with our new knowledge of some of these pros and cons. So I'd really like to thank a bunch of wonderful people that really helped us with this project. So Dr. Wade Tinkham in the Forest Biometrics Lab for all of his really gracious use of his lab equipment and processing equipment so we could get all this data processed and ready. We'd also like to thank our partner organizations, Arid Lands and Ecology IS for all their help with flight planning and project planning, as well as our funders, the National Fish and Wildlife Program and the Colorado Natural Areas Program, as well as some of the access provided by private landowners. I'd also really like to thank Teritaka Funabashi for all of his help with classifying our orthomosaics and finding all the Penstemon debilis for 2021, and everyone else who supported us along the way. Awesome, and that's what we have for you guys today. Are there any questions?
Yeah, you do have a couple in chat, but I'm not sure if you're able to see the chat, and if not, let me know.
I can't see anything in the chat, Rob.
Okay, well then I can read them to you. So Healy Hamilton asked a question I think you addressed to some degree, but it's really worth following up with. She asked, Given that you had so many known populations that could serve as training data, why did you identify your plants manually from the orthomosaics rather than using some kind of AI algorithm? And I think you're hinting at this in some of your analysis about how similar certain plants look spectrally.
Yeah, so here I'll go back to that slide just so we can have something to look at. Yeah, so she's talking about effectively machine learning and automation of trying to detect some of these plants. So this is definitely kind of where we're still moving in this direction. There's lots of new fun ways to try and get at this problem. There's the remote sensing spectral approach. There's also these new techniques, right, like YOLO V5, effectively using machine learning to look at images and classify what's in the images based on this training library. Effectively, you know, we haven't had a ton of time to dig super deep into this, but it's definitely on our radar, and we're going to try and approach both methods. So the spectral method, as well as that more machine learning, CUDA-based, you know, enhanced algorithm for looking at the actual images itself and using machine learning to detect plants. Because it takes a long time. I'm sure as Teritaka knows very fondly, it's many hours of hard work scrolling through orthos, and your wrist gets really sore. So automating it would be super great because we could just fly, throw it at a computer, have it spit out, and give us some potential locations that we could at least refine. So yeah, eventually that's the goal, but we'll see where we get.
And you're right, it takes a lot of time and effort to really look closely to distinguish. And I was going to ask a follow-up that maybe Tara would be able to answer a little better is, how do you actually distinguish some that look this similar from some of those photos? If you don't have a flowering part and the leaves are looking similar, how do you estimate your error rate?
That's a great question. You know, we're really picky about what plants that we actually decide are penstemon versus not penstemon. So for all of the stuff that we've done in this study, we've only looked at penstemon that had flowers. So that was effectively our way of saying, we're not picking up any plants that aren't penstemon. We're only looking at plants that have flowers because the flowers are unique enough in this landscape to tell us that that's a penstemon versus not. So that's how we actually kind of address this error rate. But ultimately, it's not possible for us to address error because we can't get out to some of these places to access them to go do field validation.
I heard you say repelling.
Yeah, I know you guys over in the zoology department are a little bit more fond of repelling than us botanists over here, but I don't know. I'm not as strong of a repeller, so I mean, maybe if you want to come out with us, Rob, that'd be great.
No, I'm not that strong either. I just know that Bernadette Kuhn, when she was with Botany, she had friends of hers that were rappelling into the Black Canyon looking for some plants, and so I think there's limitations with that kind of ground truthing as well. Another question.
Before we move from that topic, Rob.
Yeah.
Yeah, Neal, I thought it was also interesting how you told me that you guys have a grid on the landscape and you look in those cells until you find one penstemon that you're sure of, and then you move on. Yeah. And so when you're doing these explorations, you can't always get a count of how many penstemon are there, but you're getting you're sure that you at least have one, and then you move on to the next cell. And that's a way to increase efficiency. Yep. And the goal just being, is there penstemon there?
Right. Yeah, for our surveying. Yeah, for surveying, we're just marking one cell per each 1 meter by 1 meter grid, and it's just an occurrence. We could go and count every plant, but it takes forever. So in the sake of time, you're right, Jessica, that's definitely something that we do. Thanks for bringing that up.
And it's worth reminding people that alone is new information that's phenomenal for understanding the distribution and abundance of these plants that we haven't had access to before. So exactly. got another question for you from somebody you may know, Greg Swayze.
Oh yeah.
Would using a VNIR, I'm hoping you know what that means, hyperspectral imager provide additional information that would allow better differentiation between plants? Has this been assessed in the lab? And yeah, before you even try and answer that, explain to us what VNIR is.
So I think what he's referring to is visual near infrared. So visual and near infrared. So hyperspectral camera, forgive me if I make a mistake here, but effectively it's looking at many thousands of different wavelengths for a given pixel on landscape. Let's say we have a penstemon.
Can I interrupt you real quick, Neal? Because yeah. Greg weighed in. He said the VNIR is 0.35 to 1 micrometer.
Yeah, so it's the wavelength of light that we're looking at here, effectively, the part of the electromagnetic spectrum that we're looking at. I believe that more spectral information would really help us tease apart the flowers, most specifically. The leaves are all very similar in color, as we kind of see here. It's this very pale green. So there's not a lot of difference there. And I don't know if more spectral information would really help. But we have these flowering plants with that pink and white. I do think that more spectral information would be more helpful. And there have been studies that have started looking at mounting hyperspectral cameras on UAVs for doing this kind of work. But we just don't have access to a hyperspectral camera at this point in time. So it's a little out of our price range.
Can I ask you a follow-up on the feasibility of some of this? So you mentioned you had two cameras, the multispectral and the RGB. Are those add-ons to these drones, and how much do those cost in addition?
That's a great question, Rob. They are add-ons to the drones. So if I can scroll back here. So in the figure on the left, we actually just acquired another camera. So we'll have 10 bands total. But this camera system on this drone, so the drone's about 3,000, and then this camera system's about 10 grand. So it's about 13 altogether. Yeah, so it's pretty expensive to do the multispectral component, but the RGB is dirt cheap. The drone's like $1,500 all in all with a battery. So if I smash it on the cliff face, you know, Jill and Wade aren't gonna come after me, you know, so it's all good. Yeah, and we're insured on everything too, so yeah.
It sounds like the developments with using different kinds of cameras. I know I've been trying to investigate a thermal for understanding how bats are using cliffs. And it sounds like these drones are becoming more and more adaptable for filming different things.
Yeah.
You mentioned when you were doing your surveys, you were taking photos, correct?
Correct.
How frequently are you taking a picture while you're flying these transects, or is that automated?
So it's all manual how I'm flying the drone, but I have the drone automatically capturing an image every 2 seconds. So I'm just kind of manually, and it comes with a lot of experience at this point. But when I'm flying the drone, I have it set to capture every 2 seconds. So I'm just manually moving the drone just a little bit between each image capture. So I get this kind of representative, comprehensive photo of the whole landscape. Yeah, but you can take videos too, and you can also break a video apart into frames to do very, very similar work as well.
Okay, another practicality question is, how far can you feasibly have one of these monitor down a cliff system and still maintain control? What's the signal strength for you to be able to control that while you have the remote in your hand?
That's a great question. Multiple times when Jill and I were out there, if you're flying from the top of the cliff and you're coming down this way, usually we lose the signal about 50, 60 meters down from where we're at. And so that's just based on like, if all the radio signals going this way, it's not going down, right? Because the cliff's blocking it. So it's not perfect for this system. I mean, you could get a much stronger transmitter and really blast it out on all frequencies, but yeah, it's about 50, 60 meters down. So we can't really look that much deeper than that. But honestly, that's fine, because most of the places that we're accessing, we're trying to get really close as possible to the area we want, and then we can get to it. It also helps to come from the side as well. Jill and I found that was very helpful this last summer.
Neal doesn't mention it, but in terms of flying the drone and being that close to the ground, wind is a big factor, and there were days it was too windy to fly, and because it'll push you into the cliff if you're, 4 to 7 meters above the slope, and the slope is uneven. So those are some limitations also to doing an automated flight.
Yeah, that's true. There was a lot of sweaty palm days.
Yeah.
Very windy, sweaty palm days when I was out there this last summer.
I've got more follow-up practicality questions for you, but let's hit some more from the audience here. So Healy asks another one. How does CNHP, and this is I think for everybody, how does CNHP's monitoring program interface with habitat modeling efforts for rare plants? Are you testing distribution models with field inventory?
Yes, that is one of the next steps in this project. This is a 2-year project, and our partners on it are going to create some habitat layers from the flights that we can use to refine our existing habitat model for this species. So,
what species are next on the agenda? Knowing that you have this tool accessible, which ones have you already started planning to do this kind of surveying with?
Well, we haven't really planned for any others yet. We're still this one is one of the rarest species in Colorado. It's only known from 6 locations so far until we find some new ones. And it's got some of the most inaccessible habitat. There are a couple of other rare species that co-occur with it. So conceivably, we could try to map those as well. But yeah, it's still kind of in its infancy. And this would be real easy to use on plants that grow on flatter surfaces, like the initial example of the Physaria.
Well, Neal, you mentioned one of the factors you guys were using to try and refine your model for distribution. That was looking at the geology. Was there anything about how the slope or aspect, or I don't want to say hydrology, but anything else along those cliffs that were triggering for you all to say, all right, now we have a better idea of how we'd model distribution of this plant in other places?
Yeah, Rob, it was really eye-opening, because when I'm out there flying, I can see the shale that I'm looking after, and it has a very distinct characteristic to it. It's a little hard to describe exactly what it looks like, and that's why I tried to outline these areas in red. But pretty much all the main occurrences that occur. So Mount Calhoun and all the other areas, they occur within this band of shale. And so we just kind of follow this band of shale across the Rowan Plateau. And that's where we found new populations, actually, was in that same exact elevational gradient. Like Jill mentioned, we're working on these distribution models. We will be integrating elevation as a component into those, because we do believe that there's a very specific elevation gradient that Pensamandibulis occurs in. But I don't have a firm number for you today. So.
But you guys are uncovering features that we didn't understand before that are really going to help us identify maybe other areas where new populations could exist, just even identified by geology alone.
Right. Yeah, and our multispectral camera allows us a couple more glimpses into kind of those geological characteristics. But like my dad was mentioning, even more data, hyperspectral data would be even better for trying to pick apart some of that habitat suitability. But yeah, again, it's kind of expensive to try and apply that to this project. So.
All right, moving to the conservation realm with this information, Sarah Marshall asks, how does the number of new plants you've discovered using drones compare to the total size of the previous documented Debolus populations? How has that changed? How is your perspective on how many are out there and what's that mean for conservation?
Well, Sarah, if we're referring to what we knew before, just on the rough numbers that I have from what we've looked at, and the problem is we don't have comprehensive maps of everything, right? We're just kind of sampling spots along the landscape where, I can see clearly, and, where we were able to capture without getting winded out. And so in our biggest population, we found the most plants, and I think that was actually this site, this 1,015. I think there were about 3,000 plants that were estimated at this given population. And we sampled about 1,000, about roughly a third of that. But as you can see, we're not getting a huge part of this face. And so I think there's actually more plants. We can detect more plants using the US than were possible before with these interpolation methods, where we sample using quadrats and then we extrapolate values out. I think we're finding more plants than we were able to with that method. So.
Jill or Jessica, how's that perspective change for you guys about how many you think are out there?
Well, we've always thought there were more out there. It was just really access to them. And, these initial sites like this one here, previous people would risk life and limb to try to get close enough. And you could just really survey a limited area and then, like Neal says, extrapolate out, with binoculars, estimate based on the habitat about how far you thought it went. So I think ultimately this is going to tell us that there is more of this out there than we had previously thought. And really, we hope to find it in a few other far-flung locations. Right now, we're just finding things that are close to known sites, new locations close to the known sites, but we would like to expand the range overall of the species. And I think we can with enough resources.
So in all likelihood, we're probably driving conservation concern, not driving, but we're finding conservation concern maybe isn't as necessary because of the numbers that we're now identifying. Is that reasonable? Do we think that we're encountering more than we ever expected that are on the landscape?
I wouldn't say that yet. I mean,
Yeah.
yeah, I think that's too much to say just yet. From my understanding of what they've found, they're finding new little pockets near known locations, but there's a lot of potential for more surveys that might find more populations. But right now, right, Jill and Neal, you guys haven't expanded greatly yet,
No.
the size of these populations.
Yeah, I mean, we only added 900 this year of new plants total, 900 total, spread across a variety of sites. So I mean, I think we only have about 3,000, maybe 4,000 total that we know of for sure that have a spatial position.
For those unfamiliar with the distribution, like myself, what proportion do you think of the distribution of this plant species has been sampled?
It's a good question, Rob. I would
Yeah.
say
Good question.
5-10% of possible suitable habitat. Yeah.
That's a great indicator for that we've poorly been able to access these areas in such a way that we don't have an idea of how many are out there.
Yeah.
Yeah.
Okay, I'm going to hit you with another practical one, Neal, because I'm really fascinated by this technology for a whole host of things. It's not everybody who can just pick up a drone and start flying it. So try and explain to our audience the training that you've gone through and the expertise that you've been able to acquire and how to execute this well.
Yeah, that's a good question, Rob. So, back in 2018, when I was just getting started with the CNHP and with Dr. Michael Falkowski at the Natural Resource Ecology Laboratory at CSU, I figured it would probably be a good idea to get my pilot's license. And so everyone who wants to fly a drone commercially has to get their UAS Part 107. I think it used to cost $150. It might cost less now. You have to take an exam through the FAA, just covering like basic flight rules and all that stuff. So that's the first step. You got to get your Part 107. Second step is I spent probably about 200 hours on a flight simulator, crashing drone after drone after drone after drone until I was really starting to get more confident in flying. I also built micro racing drones for about 2 years and practiced flying micro racing drones in really challenging situations to try and get better at flying in these really precarious situations, like what I do for Jill. I think I have about 100 hours of flight time, and I think 500 flights or something under my belt right now. And so all of that is stacking up to, knowing when the wind is gusting too hard and you got to pull back, or, checking the conditions and saying, oh, we're too gusty. We can't handle it today. Like, Jill, we gotta ground it. And so it's this huge amount of time that I've spent, on my own dime, trying to get comfortable with these flying methods. And so I'm not saying everyone can go fly a drone, but doing it in this scenario, you definitely want a lot of practice. Yep.
And it's worth reminding people that this can be an inexpensive way of monitoring, but it takes a lot of the upfront time and effort that you've put into it to do it well.
Right. Exactly.
What models would you recommend for people who are out in the audience right now if they're interested in tackling this at their organization? What tools, what drones, what would you say these are things I would recommend you do besides the training you talked about?
So the things that I believe at this point in time are the best possible tools for drone monitoring, it's 3 things. So in this, on my slide here, I have a Phantom 4 Pro. They have a Phantom 4 Pro, Phantom 4 Pro V2. It's a DJI product. It is from China. There are some issues with trying to move forward with these drones in the future, but since it's $1,500 for a battery, controller, and a drone and props, everything you need to get started, I mean, it's a no-brainer. So that's the first step. You got to get a good drone. I have 10 batteries for my Phantom. So we have this full, we can roll out and fly all day long. And then we also have a battery bank in the car that we charge our batteries with too. So we're a machine when we're out there. The second thing is you have to process these imagery into something that matters, because just having 40,000 images helps nobody. Like, how can we look through 40,000 images, right? So it's stitching them together. And so software program that is absolutely renowned for this is Agisoft Metashape. You do have to get the professional version. So Agisoft Metashape Professional. The professional license for non-academic use is, I think, $3,500. The academic license is $500. So for less than, like for right around $2,000, you can have a drone and the software to process. They also have cloud processing, so you don't have to have a supercomputer to do this. The third thing, like I was mentioning, it's great to have a super powerful computer to process some of this imagery. And that's something that I've invested in recently. Beef it up, lots of cores, lots of RAM, lots of everything. And yeah, it really helps with processing some of these, couple thousand image data sets.
Did you have to have separate additional storage besides what was the computer had? Did you have to have a cloud storage facility or an extra hard drive to manage this?
Yeah, so we've stayed away from the cloud just because of the sheer volume. But right now we've got a couple different big, huge 12 terabyte drives that are hosting all of our data on a local cloud over here in the Swayze residence. So,
The Swayze data bank.
yeah.
Well, we've only got a couple minutes left, and it seems like we've hit most of the questions that have come in through here. I wanted to take the time to thank all three of our presenters, Jill, Jessica, and Neal, for taking us through this. These are some of the new technological methods I think are really valuable for us understanding what populations of rare plants and animals mean, and even communities. Their distribution and abundance are really challenging, especially for difficult to detect species like this. So Neal, we're expecting additional trainings in the future about how we might be able to use this for other organisms.
Absolutely.
So yeah, we haven't let you go yet. So everybody in the audience, I want to say thank you for attending today. Again, we'll be hosting another one of these in 2 weeks. And I think the one on topic there is some of the bat research that CNHP is conducting. But again, thank you, presenters. And I hope everybody out there enjoys the rest of the week here in Colorado. We're preparing for some slightly wintry weather in the spring, but that's just what the spring brings us in Colorado. So enjoy your week, and thank you for attending.
Thank you,
Thank
everyone.
you. Thanks, Rob.
Thank you, guys.
Bye.